The general flow of genetic information is for DNA to be made into more DNA in a process called replication. Then the DNA can be transcribed into RNA and leave the nucleus. And that RNA can then be translated into proteins that have functionality. Know the hallmarks of DNA, which stands for deoxyribonucleic acid. The DNA molecule consists of two polynucleotide strands that spiral twisted around each other. We call this a double helix. These two strands are anti-parallel, meaning they run in opposite directions, and they are held together by hydrogen bonds. And then the next is to identify some rules that we've discovered about how they bind together. So there's Chargaff's rules numbers 1 and 2. Know the difference between these two. Chargaff's rule number 1 is that A's always equal T's. The percent of A's always equal T's. The percent of C's always equal G's, meaning they're like soulmates. There are very specific binding patterns here. A's always bind to T and G's always bind to C. Okay? It took us a while to figure this out. There are multiple hypotheses before. We thought, well, maybe you can go purine to purine and pyrimidine to pyrimidine and sometimes purine to pyrimidine. But if you look at these three different scenarios, the only one that ensures a uniform width, the entire helix, is that purine-pyrimidine combo. And if you look at the adenine-thymine pair, one's a purine, one's a pyrimidine. If you look at that guanine to cytosine pair, one's a purine, one's a pyrimidine. This way, our DNA has a set width always, because it would not be very useful to us if our DNA, our genetic material, our genetic code went in, out, in, out, in, out, skinny, skinny, big, big, skinny, skinny, big, right? That sounds a lot like our unsaturated fatty acids with kinks, which made it easy to digest and break down. We don't want our genetic material to do that. Okay, so Chargaff's rule number 1 is A's always bind to T, C's always bind to G. How can you remember that? I got two mnemonics for you. One is very PC. That is the apples in the tree, the cars in the garage. Right? The non-PC one is a little bit easier to remember. Guys have cocks and tits and ass. Do whichever one helps you remember. Okay? I remember taking anatomy and physiology as a student, and my mom is the dean of, or was the dean of her nursing school. And I was struggling to remember my 12 cranial nerves. I had to memorize the 12 cranial nerves. And I was like, I don't know what to do. Do you have a mnemonic? You teach this. And she's like, yeah, I've got a great mnemonic. O, to touch, to feel, a girl's vagina, such heaven. And I was like, I did not want to hear that from you, but you know what? It stuck and I got them all right on the exam, right? And that's what matters. So apples in the tree, cars in the garage, or the other one. Rule number 2 is that the percents of A's, T's, C's, and G's are going to be consistent within a species. So let's say humans are 30% A. That makes it have to be 30% T, right? Because A's are always bonded to T. And then that leaves, that's 60. So that leaves 40% left of our 100%. So that has to be divided out to as 20/20. So let's say humans are 30/30, 20/20. That means I have 30% A's and 20% G's. And you should have 30% A's and 20% G's. And you should have 30% and 20%, and so on and so forth. There might be small, minor deviations there, but as a whole, we'd be able to round it up. It'd be like 30.01. Okay, yeah, 30%. Cool. Okay, that would be true for all humans. But Chargaff's rule takes it a step further. It says not only is there uniformity within a species, it is distinct and unique to that species. Meaning, if we were to compare ourselves to yeast, Cerebiaceae, okay, they have 32, 33. Okay, somebody rounded a little funky here because it's supposed to be perfect, right? So like 33% and 18%. That's their unique ratio, their unique mix. And then if we looked instead at bacteria, it would be Slightly different, slightly different. A dog's might be 31, 31, 19, 19. Okay, so Shargaff's second rule is that you can identify a species based upon the proportions of A, T, C's, and G's, that is unique to them. Suggesting some sort of complementary or template mechanism to fill in the gaps. There were several hypothesized models. A conservative model would suggest that the parents separated, served as templates, and then rejoined. A semi-conservative model would have this parent strands separate and each serve as a template for the new strands, which would be composed of both old and new. And a dispersive, which would chop up the DNA into a number of segments and copy different pieces. So each resulting daughter strand would be a mix of old and new for many generations to come. Messels and Stahl conducted their famous experiment on DNA replication using E. coli bacteria as a model system. And they set out to determine which of those models was the model. And so they hypothesized by using parent DNA strands that were tagged blue and daughter DNA strands that were tagged red, they would be able to discern conservative, semi-conservative, or dispersive patterns based upon the resulting color of subsequent DNA copies. What they found was in the first generation, there was a blue strand indicating that the parent DNA had been made correctly. Replication after that, it was a purple strand. And in the third replication, they had purple and red, suggesting that the second copy was a mix of blue and red. And after that, all subsequent copies contained only the new red nucleotide. Let's talk a little bit about replication, the synthesis of DNA into more DNA. It starts with double-stranded DNA, it ends with double-stranded DNA. It begins at an origin of replication. This is where the two DNA strands are separated, creating what's known as a replication bubble. A eukaryotic chromosome may have hundreds of these, and replication will proceed in both directions until the entire molecule is copied.
DNA replication begins at specific chromosomal sites called replication origins. At the origin, the DNA duplex is melted by specialized proteins, resulting in a replication bubble. DNA synthesis starts at the origin and proceeds bidirectionally. Two replication forks move outward in opposite directions. At each growing replication fork, two new DNA strands are made. The leading strand is synthesized continuously from a single RNA primer. It grows in the 5' to 3' direction, the direction of replication fork movement. The lagging strand is copied discontinuously from multiple primers, resulting in a succession of Okazaki fragments. Although the direction of DNA synthesis is opposite replication fork movement, the overall direction of lagging strand growth is toward the replication fork. As replication proceeds, the RNA primer of the first Okazaki fragment is replaced by DNA. Then the lagging strand fragments are ligated together.
So at the end of each replication bubble, a replication fork, a Y-shaped region is formed. This is where the new DNA strands will be elongating. Enzymes such as helicase, topoisomerase will assist. The helicase untwists and separates the DNA double helix. Topoisomerase corrects overwinding at the other side of the replication fork. Once open, single-stranded binding proteins will bind and stabilize the single-stranded DNA so they don't collapse in and reseal. Next in comes primase, an enzyme that can start an RNA chain from scratch. It will lay down a small runway of RNA nucleotides using the DNA template. This is known as the primer. It is the starting point for DNA elongation. DNA polymerase can attach to the primer and add nucleotides. It can move really fast, roughly 500 nucleotides per second in bacteria and 50 per second in human cells. Finally, the enzyme ligase is used to join any two fragments of DNA together, where these origin of replications fuse into one. In terms of visualizing this process, we often describe the replication complex as moving down the strand, a train on the train tracks of our DNA, when in reality, it's a stationary complex that reels in the DNA. At the replication fork, DNA elongation is happening in two directions, creating two different daughter strands. One is known as the leading strand, the other is known as the lagging strand, because it's being elongated in small segments that will then need the additional step of ligase to join them together. When describing DNA, we coin one strand the leading strand and the other strand the lagging strand. Let's see this more in detail.
In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together.
But herein lies another problem. Prokaryotes have a single circular DNA, and so it can be copied in full and completely end where it began, the entire genome transcribed. Eukaryotes, however, have non-looped chromosomes. If you unwound the DNA, it'd be one long line. And the DNA polymerase cannot add nucleotides where it is attached. So when we end replication, we have a problem. There's no way to complete the 5' ends. Because where the RNA primer falls off, there's no new primer for DNA polymerase to attach and fill in the gap, meaning that repeated rounds of replication will produce shorter and shorter and shorter DNA molecules. Nature found a way to fix this, something known as telomeres. These are special nucleotide sequences at the very end of the chromosomes that don't code for any critical genes. And so while they are shortened, they postpone the erosion of the critical genes near the ends. A special enzyme called telomerase adds telomeres in our germ cells. And the length of the telomeres determines how many times that cell can be duplicated, which gives us the average number of about 40 to 50 replications per cell before the telomere is completely eroded. It has been proposed that the shortening of telomeres is connected to aging. How do we make sure that the DNA is being copied with high fidelity, error-free? We can't. Given the size and speed, the more bases you have, the longer it'll take. But time is of the essence, because cell replication creates a very vulnerable state for these cells to be in. And for the DNA to be copied and exposed, the longer that takes, the more likely damage might occur. So there's a trade-off here. The copying of DNA must be remarkable in its speed, and there must be mechanisms in place to help proofread to ensure accuracy. It's not perfect, but the error rate is small, an error roughly 1 in every 10,000 bases. Let's examine 2 of the proofreading processes. The first is mismatch repair. This occurs during DNA synthesis. It involves endonucleases, enzymes that cut the phosphodiester bond within the nucleotide chain. That gap is then filled by DNA polymerase. It is a precision replacement mechanism. Contrast this to nucleotide excision repair. This occurs whenever damage happens to the DNA after duplication. Damage from just life, chemical or physical agents, like a sunburn. Nucleotide excision repair involves exonucleases. These are enzymes that cleave from the end and they remove a large DNA fragment from the outside of one of the DNA sequences. That gap is then filled in by DNA polymerase I, cleaning out the whole stretch. If DNA duplication has rare errors, 1 in every 10,000 bases, and the error of the proofreading mechanism is also low, another 1 in 10,000 bases, that means roughly one mistake happens every 10 million bases, but it's not zero. And so while humans are 99.9% identical, given that 3 billion nucleotide genome, there's still roughly 3 million differences accounted for through this random error and mutagenesis. But this isn't necessarily a bad thing. Remember that evolution occurs when there are sequence changes, when these changes are passed on to the next generation, and these differences allow us to identify unique individuals and circumstances. Transcription then takes the double-stranded DNA and synthesizes a single-stranded RNA message that can leave the nucleus, go out into the cell, and be expressed. Spread the news. Transcription is sometimes referred to as RNA synthesis. It's the process of making snippets of information from the DNA template. There are 3 types of RNA, and each one has its own specialized function. mRNA is the messenger RNA. This is used to communicate and pass genetic information from DNA to protein. rRNA, the R stands for ribosomal, and this type of RNA can fold upon itself. This actually forms the ribosome, which participates in translation. tRNA, the T stands for transfer. Like the ribosome ancestors, this RNA sequence will fold upon itself and become an enzyme doer that participates in translation by moving the amino acid to the appropriate nucleotide codon. The process of transcription can be broken down into 3 simple stages: initiation, elongation, termination. Transcription begins at a sequence known as the TATA box. The TATA box is a specialized area of the genetic code that's rich with adenine and thymine nucleotides. You might recall that these nucleotides bond together with only 2 hydrogen bonds, as opposed to cytosine and guanine that bond with 3. This means that that segment is easier to separate, making it more accessible to be read. There are enzymes and other proteins known as transcription factors that will bind to this promoter region to help stabilize the DNA and unwind it to make it more accessible. But the key player here is RNA polymerase. It synthesizes the RNA by following along the DNA strand and adding the complementary base pairs. So here we can see that in visual diagram. We can see our TATA-rich promoter box and the transcription factors binding to that region. And just where the start point is, we see our bubble separate those 2 strands of DNA and an RNA transcript being formed, moving along in the 5' to 3' reading frame, creating the single-stranded, newly made RNA transcript. In eukaryotes, the RNA polymerase will continue for hundreds of nucleotides past the termination sequence until it reaches a secondary encode. In eukaryotes, it needs to have confirmation before the pre-mRNA is cleaved from the growing RNA chain. I call it a pre-messenger RNA because it's not yet ready to be translated or fold up and act as a ribosomal or transfer RNA. It needs to have some modification. Eukaryotes also have post-transcriptional control, meaning that after the mRNA is transcribed, it needs some processing. It needs a 5' cap, a poly A tail, and its introns to be excised before it can be translated. There's some preparation prior to protein synthesis. The 5' cap and poly A tail help ensure longevity within the cell, while removing the introns help in speed and efficiency. Translation is what happens to that messenger RNA, and it is the synthesis of a polypeptide proteins chain that is able to actualize the instructions. Translation is often referred to as protein synthesis. This is the process of reading the mRNA message and translating the nucleotides into corresponding amino acids to create proteins that can act as transporters, communication signals, enzymes, or provide structural integrity and strength. The key player of translation is the ribosome. The small subunit of the ribosome will bind to the mRNA binding site, recognizing the start codon. The large subunit of the ribosome will bind on top of this, sandwiching the mRNA transcript. Another key player is tRNA, the transfer RNA. Each tRNA carries a specific amino acid and has a specific anticodon on the other end. And so when the units of the ribosome come together, the transfer RNA can bind complementary to the RNA transcript and deliver the appropriate amino acid. Once it does, the tRNA will leave, and an amino acyl tRNA synthetase enzyme will help match it back to its correct amino acid so it can be reused and redeliver new information. Translation begins when the small ribosome subunit and its special initiating tRNA find a start codon, a special unique nucleotide sequence of AUG. Once they sandwich, additional proteins called initiation factors will help bring the large subunit to complete this complex. During elongation, the process proceeds in a 5' to 3' direction. Binding sites within the ribosome, an A, P, or E site, regulate the addition of amino acids one by one. In each of those sites, something unique is happening. The A site stands for attachment. This is where the amino acyl tRNA will bind to deliver the appropriate amino acid. It has to match the appropriate codon and anticodon pair. The P site is where the peptide bond will form, delivering and depositing the correct amino acid. The E site is when the tRNA, now without its amino acid, will leave to come back again. Know what's happening in each site. A, it attaches. Then there's translocation. The ribosome shifts, and what was in A suddenly is now in P. And in the P spot, it will peptide bond to the growing chain. And then there's translocation. What was in P now moves to the E site, but now it's lacking its amino acid, and so it's kicked. E for exit. This will continue until a stop codon in the mRNA reaches the A site. At this point in time, a release factor, which is really a water molecule, is added. But because this is not held together by the same bonds, it stimulates the release and freeing of the polypeptide chain. A codon is 3 non-overlapping nucleotides. And because we have 4 nucleotides, A, U, C, and G, in RNA, they give rise to 20 total amino acids, 20 essential amino acids that we absolutely must have to function properly. Each codon is specific. A codon will always code for a particular amino acid. CUU is always leucine. But there's something called redundancy here, also known as degeneracy. Notice in the third base of the codon, it doesn't matter if it's a U, a C, an A, or a G. All 4 of these variations still code for that same particular amino acid. What we see is that in the genetic code, about 50% of the time, the third base doesn't matter. But there are several code words that ultimately have the same meaning. And this is due to a phenomenon known as wobble fit, that the third base doesn't sit as well during our complementary base pairing of the codon and anticodon. And so over time, the ribosome has evolved its own set of rules, and it will almost always exclude or ignore the third base in the anticodon pair to prevent massive missense mutations occurring. And so we see in this table showing the codons and their appropriate amino acid codes that there is a lot of redundancy, multiple ways in which you can code for the same amino acid. Translation is often referred to as protein synthesis. This is the process of reading the mRNA message and translating the nucleotides into corresponding amino acids to create proteins that can act as transporters, communication signals, enzymes, or provide structural integrity and strength. The key player of translation is the ribosome. The small subunit of the ribosome will bind to the mRNA binding site, recognizing the start codon. The large subunit of the ribosome will bind on top of this, sandwiching the mRNA transcript. Another key player is tRNA, the transfer RNA. Each tRNA carries a specific amino acid and has a specific anticodon on the other end. And so when the units of the ribosome come together, the transfer RNA can bind complementary to the RNA transcript and deliver the appropriate amino acid. A codon is 3 non-overlapping nucleotides. And because we have 4 nucleotides, A, U, C, and G in RNA, they give rise to 20 total amino acids, 20 essential amino acids that we absolutely must have to function properly. Each codon is specific. A codon will always code for a particular amino acid. CUU is always leucine, but there's something called redundancy here, also known as degeneracy. Notice in the third base of the codon, it doesn't matter if it's a U, a C, an A, or a G. All 4 of these variations still code for that same particular amino acid. What we see is that in the genetic code, about 50% of the time, the third base doesn't matter, but there are several code words that ultimately have the same meaning. And this is due to a phenomenon known as wobble fit, that the third base doesn't sit as well during our complementary base pairing of the codon and anticodon. And so over time, the ribosome has evolved its own set of rules, and it will almost always exclude or ignore the third base in the anticodon pair. And so we see in this table showing the codons and their appropriate amino acid codes that there is a lot of redundancy, multiple ways in which you can code for the same amino acid. Once it does, the tRNA will leave, and an amino acyl tRNA synthetase enzyme will help match it back to its correct amino acid so it can be reused and redeliver new information. The 3 stages of translation are very similar. There's an initiation phase, an elongation phase, and termination phase. All 3 stages require protein factors that will aid in the process. Translation begins when the small ribosome subunit and its special initiating tRNA find a start codon, a special unique nucleotide sequence of AUG. Once they sandwich, additional proteins called initiation factors will help bring the large subunit to complete this complex. During elongation, the process proceeds in a 5' to 3' direction. Binding sites within the ribosome, an A, P, or E site, regulate the addition of amino acids one by one. This involves elongation factors, codon recognition with complementary pairing, a peptide bond being formed, and then the shifting of the ribosome, translocation down the transcript. In each of those sites, something unique is happening. The A site stands for attachment. This is where the amino acyl tRNA will bind to deliver the appropriate amino acid. It has to match the appropriate codon and anticodon pair. The P site is where the peptide bond will form, delivering and depositing the correct amino acid. The E site is when the tRNA, now without its amino acid, will leave to come back again after it has been fused with a amino acyl tRNA synthetase. Know what's happening in each site. A, it attaches. Then there's translocation. The ribosome shifts. And what was in A suddenly is now in P. And in the P spot, it will peptide bond to the growing chain. And then there's translocation. What was in P now moves to the E site. But now it's lacking its amino acid. And so it's kicked. E for exit. This will continue until a stop codon in the mRNA reaches the A site. At this point in time, a release factor, which is really a water molecule, is added. But because this is not held together by the same bonds, it stimulates the release and freeing of the polypeptide chain. Recall that a mutation is a change to the genetic material, and this can occur within a cell or virus. Mutations can affect one nucleotide or several nucleotides, and sometimes that change can affect the overall protein structure. Given that form dictates function, it can have lasting downstream effects. On the bottom, we see an example of where a single nucleotide change in the DNA template can lead to the production of abnormal proteins, healthy red blood cells, into sickle-shaped red blood cells, unable to carry oxygen. Point mutations are caused by nucleotide pair substitutions, where one nucleotide is replaced by another one. The results can be silent, missense, or nonsense mutations. Know the difference between these three. A silent mutation changes the sequence of the genetic information, but it's considered silent and not harmful because the overall amino acid has not changed. This is due to redundancy in the genetic code. Sometimes, however, that substitution will result in a new amino acid being added. This is termed a missense mutation. If the new amino acid happens to be a stop codon, you get a nonsense mutation. Nonsense mutations nearly always lead to a non-functional protein that will be degraded and reused, but can be problematic because it has utilized resources and delayed the replenishment of a much-needed protein. Taking a look at each of these examples now in visual form. Here we can see a single nucleotide substitution, an A instead of a G, which then leads to a U instead of a C being incorporated. But because the wild type and the mutated strand both add a glycine, it is a silent mutation. In this example, a thymine has replaced a cytosine, and that leads to the mRNA transcript having an A instead of a G. This codes AGC versus GGC, and that changes which amino acid was added, a serine versus a glycine. This is a missense mutation. In this last example, adenine replaced thymine, leading to a uracil in the mRNA transcript, and UGA codes for a stop. This sequence is utter nonsense because it literally was one amino acid and ended instead of continuing on the code and adding the lysine, the phenylalanine, and glycine beyond. Any additions or losses of nucleotide pairs in a gene may alter the reading frame, producing a frame shift mutation. This is missense on high. It doesn't just change one amino acid, but every amino acid after the fact. The example here is the fat cat set, common English words that follow the three-letter codon pattern. Let's say we delete the T. There is no space saving amongst codons. What happens is everything shifts down. And so then we only get that first codon that made sense, the. After that, utter gibberish. Every amino acid after the addition or deletion being altered in some way, shape, or form. So here we see that depicted. A single A deletion results in a U deletion, and that changed phenylalanine to leucine, but more importantly, glycine to alanine, and so on and so forth. Where do these mutations come from? Many of them are spontaneous. They just happen, errors during DNA replication, recombination, or repair. But sometimes they can occur more frequently due to mutagens, physical or chemical agents that damage our DNA. And so recognize the specialization. DNA stores genetic information. RNA sends messages, translates it, and proteins do the work. Why can't DNA just go straight to protein? Why did evolution favor this specialization? That RNA intermediate provides protection for the DNA and allows more copies of protein to be made simultaneously, since many RNA transcripts can be made from one gene, and each gene transcript can be translated repeatedly over and over again. The summary is much more accessible than the entire book.